trying to separate it out from all the other proteins in the cell. We went through these different chromatographic techniques that are based on different properties of proteins. But then, as I presented the chromatographic techniques, I always talk about detecting the proteins coming off the column as it is the column that's being eluted. And so what we need to do is talk about different ways of detecting proteins. And there are general ways of detecting proteins. And one is to take advantage of the absorbance spectra of the aromatic amino acids, particularly around this 280 nanometer region. And again, the only reason for not, you actually have a higher absorbance down at the low values, but the reason for staying away from there is almost anything that's in your buffer or whatever absorbs down there, so it's not very specific to proteins. But 280 can be somewhat specific to proteins. And then so that you end up getting a typical protein UV spectrum that looks like this, with a maximum around 280 nanometers, and usually sometimes you'll see nice tryptophan flavor in there. All right, that's not the only technique. There's also, you can analyze by Kamasi brilliant blue binding. That's a it's also known as the Bradford reagent here. And so this is the structure, and what it basically does is it binds basic aromatic, or basic amino acid residues. And what it does, it causes a shift in absorbance at 595 nanometers. So, again, let's see, here's the structure of Kamasi blue here, again. And then, when Kamasi blue is unbound to protein, it has this peak here, this A peak here, at the maximum amount 500, and then it dips down at about 595, and then it comes back up. However, when it binds the protein, it shifts, and so that you end up actually getting increased, greatly increased absorption at 595 nanometers. So basically, by monitoring binding at 500, absorbance at 595 nanometers, you can follow the elution of protein off of the column. This is also typically used to stain gels. We're going to talk about SDS page here in just a second. Yeah, actually, let me, before we do this, get our outline going and introduce this a little bit. Okay. So we talked about absorbance 280 nanometers to the aromatic amino acids. The second way of detecting total protein is, just to get it down, is the Bradford assay. And this finds Kamasi dye which causes a increase in the absorbance at 595 nanometers. Again, this binds, Kamasi dye binds basic and aromatic residues. All right. Now the other way you can detect proteins and get a little bit more information, these two methods we talked about so far are just about, detecting whatever protein comes off. But you can get a little bit more information by actually analysis by SDS-PEG. By an SDS-PEG is sodium glutesyl sulfate fancy names for dish detergent, polyacrylamide gel electrophoresis. And this separates by the molecular weight of the protein. And so electrophoretic technique is similar to agarose gel electrophoresis, where we talked about separation of nucleic acids, in which you basically put a protein in an electric field, and it will migrate based on its charge. Now, the problem with proteins is we've talked about proteins as variable pI. So at any given pH, okay, so at any given pH, we different proteins will actually have different charges, right? So at pH 7, the histones, which are positively charged, would move in one direction, whereas, I don't know, another protein, most proteins are negatively charged and they move in another direction. Now, to basically wipe out, cancel out that variability in charges, you find the SDS. And what the SDS does is it provides a constant charge to mass ratio. So if here we have our protein, this is our native protein. Now I've linearized it. Maybe it has some positive and negative charges on it, positive, negative charges on it. Depending on the relative amounts of those positive, negative charge, it'll either have a net overall positive or net overall negative charge. Okay, but what SDS does, just a reminder of the structure of sodium dodecyl sulfate. Got a hydrophobic tail, and then a doubly negative charged sulfate group. It basically binds to the polypeptide backbone and then provides a negative charge. And that negative charge basically swamps out any of the native charge that's on the protein. So now, the SDS binds the protein and provides constant charge per mass ratio. So that the smaller proteins end up moving faster because they can make their way through the matrix, and the bigger proteins move slower. The other thing the SDS does, SDS also denatures, that is, linearizes the protein. What this does is remove any shape effects. You know, because if you had one protein that was in a a nice compact little ball, and you had another protein that was kind of long and skinny, maybe more rod-shaped, like they would migrate differently through the matrix. What the SDS also does is basically unfolds the protein and gives them all the same shape. Now, unfolding the protein, though, also remember that a lot of proteins here's our amino terminus, maybe here's our carbon. carboxy terminus of the protein. The structures are stabilized by disulfide bonds. So also to linearize it, you have to reduce these disulfide bonds, break those disulfide bonds so that it can unfold. So to do that, you treat with beta-mercaptoethanol. Beta-mercaptoethanol is and what that does is reduce these disulfide bonds. Now you and basically the beta-mercaptoethanol becomes oxidized. Beta-mercaptoethanol is probably the most common reductant used to break those disulfide bonds. It has a particularly unpleasant smell. So some people don't like working in biochemistry labs because there's always beta-mercaptoethanol, or I used to call it, it's the smell of biochemistry. So, but there's another molecule called dithiothreatol that works pretty well. It's more expensive. So fundamentally, what you then is you have a gel made out of polyacrylamide. There are wells on top of the gel. I'll show you a picture of an apparatus in a bit. And then we have we put in our protein. That has been reduced with beta-mercaptoethanol. Beta-mercaptoethanol is often abbreviated BME. And so it's all in a mixture at the beginning. You apply a voltage to it. It migrates towards the positive charge, and basically the protein gets separated out based on some molecular weight. And typically you'll have a standard with known molecular weights that you can then compare to. And so if you started the little exercise for Wednesday, you kind of go through this. And certainly the folks in 162 have already seen this kind of thing. So maybe you might be analyzing different fractions or something, and so you'll get different proteins. apparatus in here, and you're loading the samples up here, and while it's in the field, the migration is going to go in that direction. And then this is a gel. And the first lane here are your molecular weight standards. And you can, the matrix, the gel matrix is polyacrylamide. And you can adjust the concentration of polyacrylamide to separate out at different size ranges. So if you have big proteins, you have a lower percentage of acrylamide. If you have small proteins trying to separate, you'll have a higher percentage of acrylamide. And then this is just one particular protein, carbonic anhydrase, where they just added more and more of it, because you're going to see. So it's got a molecular weight of around, I don't know, 30 or so. All right, before leaving this whole detection thing, though, I do want to talk about a couple of other methods for detecting specific proteins. So, you know, the A280 and the Bradford, they just detect total proteins. This method, say you're analyzing fractions coming off the column, at least tells you the size of the proteins that are eluting from the column. But then the next method is, what if we're looking for, say, a particular enzyme? So take hexokinase, for example. The first enzyme of glycolysis takes glucose and phosphorylate to make glucose 6-phosphate. So it's a protein that serves as a catalyst. We're trying to purify that. And, you know, we may even know its size. We may know it's about 60,000, molecular weight is about 60,000. But even using SES-PEG, there might be so many proteins in there we can't actually detect that protein from any of the other ones. So the question is, well, how then do we detect where our hexokinase is? And so what you do is you do an enzyme assay. And you basically take each fraction and look for the ability to catalyze the reaction. So you might take a mix up, you know, if the enzyme is hexokinase, it takes glucose and ATP and makes glucose 6-phosphate and ADP. So, and these reactions, like most enzyme catalyzed reactions, don't proceed at any rate in the absence of the catalyst. So if you mix glucose and ATP together in a test tube, nothing happens. Okay, but if you if you add the catalyst, then the reaction will go. We just take a little bit of each fraction, you know, set up a whole bunch of tubes with glucose and ATP in it, take a little bit in each fraction and say, okay, which fraction actually results in the production of the product? Which fraction actually has the catalyst in it? So you can kind of blindly follow the catalytic activity, not necessarily being able to see the protein on a shell at all. And, you know, so often when you do this, you look for the appearance of a product. Or sometimes you follow it. It depends on what you're looking for. Sometimes it's easier to follow actually the disappearance or of a reaction. Now, another way a final way of looking for a specific protein is to use an immunoassay. Which is also known as a Western blot. And here, it requires some prior work. It requires that you maybe purify the protein before, and then use that protein to generate antibodies against that protein. Antibodies being proteins that specifically recognize your protein of interest. And then there are colorimetric reagents that you can link to those antibodies to be able to visualize. You basically react with antibody raised against protein of interest. And then you visualize the antibody and there's several different methods, but visualize the antibody, I'm going to say, using a variety of colorimetric reagents. And you do this, you do a Western blot in 162. Although it's a little bit funky, that one's a little bit funky because we actually do work it with lectins, but it reacts against carbohydrates. But the basic idea is there. All right, so this is it. So this is kind of the end of the protein purification. We talked about the problem, we talked about the different methods that we actually use to separate, and then finally the different ways of actually detecting the protein. Now I want to finish up this sort of technological section and talk about amino acid analysis of proteins. You know, we talked about what we've introduced so far with proteins is its primary structure, the sequence of amino acids of a protein. The question becomes, well, how do we know what the amino acids are in a protein, and particularly, how do we know the sequence of amino acids within a protein? The first thing that's often done is just say, what's the total amino acid composition? And let me just kind of put everything a little bit of historical perspective. Prior to the genomics era, where there was a tremendous increase in the volume of DNA sequencing that allowed us to identify the DNA sequence that encodes a protein, maybe before we've even never purified it or analyzed it. Before the genomic era, basically, you started by studying the protein, and then you work backwards towards getting the DNA. So you might do a lot of work to purify a protein, and then do things like study the amino acid composition and ultimately sequence the protein. So a lot of these methods I'll be talking about are used less because if you know the sequence of the gene, well, you can just use the genetic code, figure out what the amino acid sequence is, and you get your composition and all of that. Although it's still used for a variety of reasons. Things like amino acid composition are important in nutritional studies. You know, if you've got a particular diet, you want to know what the amino acid composition is in a particular diet. You do a lot of amino acid composition work. So there's still uses for these particular techniques, and there's a lot of methods out there for developing these techniques. So if you want to know all the overall amino acid composition without necessarily knowing them in order, the first thing that you do is you have to hydrolyze the protein. Take the protein and break it up into individual amino acids. Hydrolyzing a protein, you're adding water across that peptide bond. And again, I've mentioned this before, this is pretty harsh treatment. 6 normoid Cl, 120 degrees C overnight. But now you're taking your protein and you basically cut it up into individual amino acids. What's that? Yeah, I don't know. That's just that's the standard way of doing it. You have to do it in a sealed vial because at 120 degrees it'll boil. So you have to, it's under a little bit of pressure too. Then the next is that you derivatize it. And we won't go through the methods, but basically, if you separate out the amino acids directly by HPLC, one of the difficulties is it's difficult to actually detect them coming off an HPLC column. And so you typically would derivatize them, have them something either react with the amino part or the carboxy terminate that adds some sort of tag on it that allows you to detect the protein. So you basically derivatize and analyze by reverse phase HPLC. And honestly, most of this stuff is all automated. So you basically stick your sample in, and it'll do a digestion, and a little robot will move things around, so and it comes out. You take a chromatogram, and it quantifies everything for you. So this tells you, okay, what are all the different amino acids within a protein? The next thing you might want to know is, well, how big is your protein of interest? Is it 100 amino acids, 500, 1,000, 10,000? And we talked about one way of doing it, and that's use STS page. Just run it on, if you have it purified, you just run it on a gel next to a bunch of molecular weight markers, you can figure it out. That's kind of the low-tech way of doing it. It's still used pretty often. The more up-to-date is to use ESI as electrospray ionization mass spectrometry. And this is basically a gentle way of ionizing your sample, so your protein, so it doesn't fragment and doesn't break up into different fragments and gets different charges. You usually get a family of different peaks, but it's basically a good method for getting the total size of the protein. All right. So this tells us what amino acids are there, how big it is. Now what we really want to know is what the sequence of amino acids is. Now, the sequencing technologies, there's an older technology called the Edmond degradation, and then there's some mass spec technology. Both of them, though, sort of have an upper size limit to how big of a fragment that they can actually sequence. So usually what you have to do is actually chop your protein up into smaller fragments, and then they analyze the sequence of separate and analyze the sequence of each fragment. Okay? So the first step is to generate fragments that can be sequenced. And the first thing you need to do is, just like for SDSP, you have to treat with beta-mercaptoethanol to break the disulfide bonds. And the other part about this is you'll also, after you break the disulfide bonds for sequencing, you treat with iodoacetate to derivatize the cysteines. The cysteines don't sequence particularly well. To make carboxymethylcysteine. No, I'm sorry, I got that back. All right, so you basically broken those disulfide bonds, treated the free thiols that you generated to generate this thing that can be sequenced better. Okay, then the next step is to treat with proteases to generate smaller fragments. And so proteases that you typically use include trypsin, which cleaves after basic residues. And one of the reasons it's important to actually cleave with two different proteases, or use two different methods to cleave to proteins, is because your trypsin, you're going to generate a bunch of fragments, and you can sequence those fragments. You don't necessarily know the order of those fragments, though. Whereas if we're treating with chymotrypsin, you're going to get a second set of fragments, and you get the sequence of those fragments, but you don't necessarily know the order of those. But by looking at, by combining the two, you can basically stitch together the order of the sequences. All right, so trypsin is a common one. Chymotrypsin which cleaves after aromatic amino acids. And then there's another method of cleavage that is actually not proteolytic, but it's a chemical cleavage. With cyanogen bromide. This cleaves after methionine. This one's good for getting big fragments because there usually aren't that many methionines in the protein.
So would you say like 2 is ideal, or say it would be better to cleave with 3 instead?
Oh, my guess is what you would usually do is do a couple and then see how it looks, and if you need some clarification, maybe do another set. All right, now you've got these fragments, maybe 20 to 30 amino acid long fragments. And now you can separate those fragments by HPLC and then sequence each fragment. So okay, the classic technique for sequencing the fragments is the Edman degradation. And I'm holding myself back here because up in time till this time around, I always go through and go through the chemistry of the Edman degradation, how that works and how it ends up cleaving off of that. But the reality is, if this technique is kind of outdated, nobody uses it anymore, so it just would be just kind of an exercise in organic chemistry. Okay, so I'm not going to actually go through the Edman degradation this time around. Your textbook doesn't go through the Edman degradation, but rather we're going to refer to the more modern technique, which is tandem MSS. And here, the proteolytic fragments are ionized and selected by size. So the fragments, you basically, you treat it with your proteolysis, and now you have a solution for a whole bunch of little peptide fragments. You do a soft ionization first, basically, that doesn't break up those fragments, but you can go through and you can ionize them and select them based on a particular size. Say, okay, I only want fragments that have a molecular weight of 1,300 or maybe 10,000 or something along the line. Then the second ionization is the second ionization. So this is the first ionization. Then the second ionization generates subfragments are generated. So that fragment that is, say, 10,000 then gets subject to a stronger electromagnetic field that actually fractionates that, more like traditional mass spectrometry, and then those individual ions are quantitated.
Sorry, what kind of cell is it? Collision cell?
Collision cell. It actually collides with ionized argon gas. I'm not going to go into that. Now, this is what really basically will allow this method to become the standard method. Is now you say, okay, here's a peptide fragment that is a molecular weight of 10,000. Now, when I zap that fragment and break it up into a whole bunch of different fragments, here are the different size fragments that I get. And what is really allow this thing to work is the deconvolution, basically the computer power to look at those fragments, deconvolute all those fragments and say, based on those fragments, this is the amino acid sequence. That you basically get, I'll show you a little bit, but you basically get a collection of a whole bunch of different fragments of different sizes, and by careful analysis of those sizes, if you had to do it by hand, it would just, it would take you a year, right, to figure it out. But with the computer power, it basically, you can go through and you can analyze the different fragments that are generated, deconvolute that, and it'll tell you what your sequence is. And so the final thing is computer analysis of fragmentation pattern to deduce the sequence. All right, the final thing I'm going to just mention is, I already alluded to it, is you get the sequence of all these subfragments here generated by your proteolysis, but you still have to assemble those. You have to figure out the order. So the final step is assembly of fragment order. And again, what you do is use alternate cleavage methods and then use one set to order the second set. And I believe you have a problem in your in the homework that basically has to go through and do this. It's pretty straightforward. All right, let me All right, so here's just a picture. First of all, the total amino acid analysis, this is what one of those things looks like. I think what this meant is all automated, you took your sample and it works, developed the chemistry behind it. But we didn't talk about Dantzler. The other thing you can do is you can figure out what the amino terminal, if you just want to know what the amino terminal amino acid, you can react it with this reagent, dimethyl chloride, and you just get the amino terminal. That's not done very much anymore. We won't talk about this. This is the Edmond degradation. This is reacting with phenyl isocyanate, go through all of this sort of stuff. And again, I'm wistful, you know, walk through this chemistry. The Edmond degradation also was pretty automated. Basically, here's an example of a peptide sequence. Okay, but then this is the tandem MS and MS, the peptide sequence analysis. You have your sample that has a bunch of different peptide fragments in it. You ionize it, then you separate those based on the size, and then you collide them to take those separated fragments and further fragment them, and then you separate them, and then you detect, and then you have to do the deduction of the sequence. And so this is sort of the complexity of what we're going on. So say we have this particular sequence here, okay? What happens is, during that second fragmentation, it'll cleave like to the C-terminus of here, generate a whole series of fragments, and then it'll cleave on the N-terminus here, generate a whole another set of fragments. And so you get this really complex spectrum in here. These different letters reflect whether they're the N-terminal cleavage or the C-terminal cleavage. And it's basically the ability to take all these, and this is our size down here, take all these different peaks, all these different ion abundances, and deconvolute it into a particular sequence that allows you to get that. All right, we'll finish up there. We're gonna